All right, let's get started. So I'm going to finish the test two review sheets with you. And at the end, if we have time, and you guys have like additional questions, we can certainly go through those as well. So I hand this sheet out actually, I did not hand it out. I posted on eCentennial for you last time. So, if you haven't already got a copy yourself, please, get one as soon as you can. And if you miss the things that we did last class because you weren't there, I will post this whole thing on eCentennial afterwards as well. But let's pick up where we left off last Wednesday. This is question number 3 in the protein synthesis section. So here we go. Hemoglobin is an oxygen-carrying protein found in red blood cells. Its quaternary structure, so level 4, right, contains 2 alpha chains and 2 beta chains. Thalassemia is an inherited blood disorder that causes your body to have less hemoglobin than normal. In some forms of thalassemia, the beta chain, this is the key phrase here, the beta chain are completely missing in some hemoglobin. What kind of mutation is likely cause of this? Explain. Well, if you are missing a whole chunk of hemoglobin, like an entire chain here, there's only one mutation that is capable of doing that, and that would be the nonsense mutation. Okay, so this is likely a nonsense mutation, right? And what is a nonsense mutation? Now I'm just going to tell you things that you should know about nonsense mutation. Nonsense mutation is it can happen in 2 ways. Yeah, you can have base substitution, okay? Creating a stop codon. Okay, so very simple. If you have UAC and then it becomes UAA, that is a nonsense mutation. And you're doing this by changing a single nucleotide in this case. But there is also another way for creating nonsense mutation. Okay, what is the other method of creating nonsense mutation besides base substitution? How else can we cause the nonsense mutation? That's right, you can have insertion or deletion. Insertion or deletion, right? Where the new reading frame contains a stop codon. Okay. So it could be either one of these situations. There's no way you can figure that out based on the question. But the fact that you're missing a whole chunk of hemoglobin tells you that it has to be some kind of nonsense mutation. All right. I guess we can explain a little bit further by terminating translation prematurely. The protein is truncated, right? We say. Or in some cases, completely missing. Right? If the stop codon occurs very early in the sequence, then you end up missing the whole protein. If it happens halfway, you lose half of it. If it happens in the end, maybe it's okay. Right, so that takes us to the next question. Explain the likely outcome of missense nonsense and frameshift mutation. So missense, let's just write missense. It would have the greatest effect if it happens in a crucial part of the protein. Example, the active site of an enzyme. It depends where it happens, right? If you do the missense in the active site of the enzyme, you will change the shape of the active site, and then the enzyme might become non-functional, right? Also depends, right? Severity depends on the type of missense, right? Some amino acids have similar properties. So like if you have an amino acid that's like hydrophobic, and after the mutation it's also hydrophobic, then you would have less of an effect, right, on the protein compared to if you're switching something from hydrophobic to something that's hydrophilic. Okay. If the missense mutation changes the amino acid to something with similar property, right, e.g. hydrophobic to hydrophobic, and the impact is minimal. Okay, so number one depends on where it happens. And number two depends on what kind of substitution are we talking about. It's not always bad. Okay? It's possible you have a substitution with something that is similar enough that it will not cause a big problem. All right, we talked about nonsense already earlier. I don't think I'm gonna write it out. I'll verbally tell you again. Nonsense, it depends on where it happens, right? If it happens at the beginning, it's the worst. If it happens at the end, it's probably okay. That one's easy to understand. Right? Frame shift. Let's quickly talk about frame shift. Frame shift. Okay, something is wrong with the program. Give me a second. Let me see if I can fix this thing. What is this thing doing here? Okay, let's try again. Frame shift. Right. Frame shift is when you insert or delete nucleotide. Right? Not multiple of 3. then what will happen is the reading frame of codons shift. So that will lead to extensive missense mutation, right? Or if there is a stop codon in the new frame, it's possible to create nonsense mutation as well. Okay. So is it bad? Again, you have to think about where it happens, right? Beginning, probably bad. Towards the end, it could be bad as well. Okay. Like if I have to choose between a frame shift that happens at the end versus a nonsense that happens in the end, theoretically, the nonsense towards the end is better than the frame shift happening at the end. Here's a reason. If you have the frame shift happening at the end, that means you are shifting the reading frame at the end, which means the original stop codon is going to be gone in your new frame, right? So yes, you make your normal protein, but then because of the frame shift at the end, you will not stop where you're supposed to stop. And then you keep on going and you have this big chunk of like extra stuff that's attached to your protein, right? And that might interfere with how the protein works. Okay. So again, just for the sake of argument, right? Theoretically, if you have a nonsense mutation towards the end of a protein, that's probably better. Less, when I say better, I mean like less impact compared to a frame shift that happens at the end. Okay? But if it happens at the beginning, they're equally bad. And nonsense at the beginning is as bad as frame shift at the beginning, I would say. So that's that. Any questions? You can just type the question in the chat, all right? If you have any, or you can unmute and talk to me. That's okay as well. All right. Even for online, I only have like 1/3 of the class. I thought people would be like it would be easier for you guys to come. Apparently not. Okay, so number 5, use a diagram to illustrate the following terms: the promoter, the intron, exon terminator, so on and so forth. So very briefly, we can go through this again. You have your gene here. Okay, so this is your DNA, okay, which contains the gene. And the gene is marked by the beginning sequence. Yeah, beginning sequence right here, which we call promoter. Okay, it marks the beginning of a gene. And it's AT rich. We know that already. And then at the end, we have the terminator sequence. Okay, at the end here, where you want to stop the transcription, you would have to have the terminator sequence. End of a gene. And of course, you would have like the rest of the DNA, right? Like the parts that we don't need, right? We're not copying them. They're not needed at the moment. And then we're gonna go transcription. okay, which includes 3 steps, right? We have the initiation, elongation, and termination. And this is done by RNA polymerase. Okay, so you don't have to describe the process, but you should be aware of what's happening. Okay, so there's the template strand, there is the coding strand that we talked about, and in the end, you're gonna get this RNA transcript, right? Okay, so the RNA transcript, this is pre-mRNA. Okay, it's not ready yet. And the reason it is not ready is because when we copy the genetic material, it turns out there are some like non-coding DNA, right? These are non-coding DNA that needs to be removed. And I hope you remember the non-coding DNA, right? These are called introns. Right? They are non-coding. Whereas everything else in between, those are the coding ones. And we have a name for them as well. Those are going to be the exons, right? Okay. So the question is asking us to explain intron, exons, terminator, promoter, and then the poly A and the G prime cap. Okay. So this is 5' to 3'. To stabilize the structure, you would add a G prime cap over here, G cap, and then we would add a poly A tail, right? This would increase the stability of the RNA transcript, so it can stay in the cytoplasm for longer periods of time. Okay, so now you have this, and then you have the G cap and the A tail. All right, and all the introns are going to be removed, and you only have the exons. Okay, so this is like called RNA splicing. Right? To splice is to cut and connect. It's like doing arts and craft. Okay, that is that. Let's take a look at the next one. Explain the relationship between codon, anticodon, and tRNA and amino acid. You should know this already. I wrote it down for you several times, but let's do it again. Let's say we have the DNA template, the DNA template strand. Let's say it's ACTG. Then the DNA coding. I mean, you really should know this by now. Okay, there are several questions in the multiple choice for this. You are not going to have to do transcription translation in the written portion of the test. Okay, these things I'm going to be testing you in the multiple choice. So I'll give you like a sequence, and then I can ask you, what is the template strand, or what is the RNA sequence, or what is the protein sequence, right? So that's where I'm going to be testing you. So of course, template strand and coding strand, they're complementary. And when you do the messenger RNA, it is going to be the same as the coding strand. Okay, and then the anticodon. Okay, I should say tRNA here. So this is the codon, and this is the anticodon. That's where it's found. Okay, and that would be ACUG. Okay, so I'm just going to highlight the stuff that are the same, and then I'm going to highlight the things that are complementary to each other. Okay, so template strand is actually same as the tRNA, and then these things are the same as well. Okay. So different colors are complementary to each other. Same color means it's identical. But of course, you replace the T with the U, right? All right. So pretty much you have to know this. And that's it for this lecture. Does anyone have any question before I move on to heredity? Okay. Next, explain the significance of crossing over. How, why, where, when does it happen? Okay. So crossing over is the exchange of genetic information between homologous chromosome during prophase 1 of meiosis. Right? That's the what and the when. Now you might remember these pictures that we drew in class. You have the chromosome 1 from mom, and then you have chromosome 1 from dad, as in like your mom and your dad, right? And this is meiosis happening in your body. Okay. So let me just reorient you. These two are sister chromatids. They are identical copies. Okay, so there is no point of doing crossing over between the sister chromatids because they are exactly the same. If you swap them, it will look the same afterwards. So there's no point of doing that. Okay, so this here is called a tetrad. Right? The homologous chromosome finds each other during prophase 1. Okay, so right here, the blue one and the red one, these are homologous. And in case you forgot, homologous are the same size, same length. They have same order of genetic information, but the version might be different. The version of the genetic information might be different. Big A, small a, right? So afterwards, what's going to happen is you will have the exchange of material, and you will end up like, let's maybe draw it properly, okay? Like you actually going to have the crossing over that's happening all over the DNA, okay? And I know in class I just showed you that there is a little bit of blue at the bottom, but I really want you to understand right? This process is happening throughout the entire chromosome. Okay. So like in the end, you have like this chimera, right? Like this blend of red and blue. Okay, so the purpose, like why are we doing this? This is to increase genetic variation in gametes. Okay, and where is it happening? Well, it's happening in the gametes, right? Yeah, sorry, in the germ cells. Happens in the germ cells because only germ cells can do meiosis, right? And in meiosis, you create genetically different gametes. That's why we have this recombination going on. You don't want this recombination happening in your bodily cells. Right? In mitosis, we want to create faithful copy of the original. Okay, so that is crossing over. What is the purpose of meiosis? Well, purpose of meiosis is the basis of sexual reproduction. Okay. Where does it happen? So germ cells and germ cells in the gonads. Undergo meiosis to create genetically diverse gametes. Gametes are egg or sperm, right? And the cells, the gametes. Okay, I should probably put this down for you. The germ cells. Oops, it's still very thick. Give me a moment. The germ cells are diploid. Two sets of chromosomes, right? But the gametes, they are haploid. 2N and N. Okay? That is meiosis. Okay, non-disjunction. Anybody remember the word non-disjunction? Like in one simple sentence. What is it? The result is extra chromosome. That's correct. But like what causes it to have the extra or missing chromosome? That's right. In essence, non-disjunction is when chromosome do not separate. During anaphase 1 or 2. Okay. Consequences? Gametes with extra or missing chromosomes. Okay. I mean, it depends when it happens, right? If it happens in an anaphase 2, then you would have half the gametes being okay. And then one would have extra, one would have missing. But if it happens in meiosis one, during anaphase one, then all the gametes is going to be problematic. 50% will have extra, 50% will have missing. Okay, so that is non-disjunction, when the chromosomes don't separate. Practice on all the different types of genetic problems: monohybrid, dihybrid, codominance, incomplete dominance, sex-linked. So like I mean, I don't have a particular one I want to go through with you, but you should definitely do it yourself. I mean, we could I can make one up. We can do it. Can I explain mono? Mono means one trait. So like, you know, brown eyes and blue eyes. Okay? And then you're only doing like big B, small b. Dihybrid is like when you do two traits, right? So let's make something up, okay? So let's say we have mouse here. Okay, mouse. Mouse 1 here, and then mouse 2. And let's say this one is a white and long tail. Okay, and then over here, we have a second one. And this one is going to be gray. Gray and short tail. Okay. Let's say this is the P generation and they are purebred. And then the F1 generation, 100%. 100% looks like this. Okay. 100% is gray and long tail. So what does this tell you? What information can we get based on this result alone? That's right. Based on this alone, you can conclude that gray is dominant over white and long tail is dominant over short. Remember, we wrote this down like several times in class. And I said when the parents are purebred, all the F1 generation will only possess the dominant trait, right? So even though I don't tell you in the question which one is dominant, which one is recessive, you just look at the F1 generation. Since all of them are gray and long tail, they must be dominant, right? The gray and the long must be dominant. Okay, so let's say we same mouse here, okay? But let's consider you know, big G, small g, big L, small l, cross with a small g, and a big L, small l. Okay, so I'm going to give you a moment to figure this out. Okay, do the Punnett square. Right, and let's say we have I don't know, 100. That's too easy. Let's do you know, 750 mice. Okay, how many do you expect to be gray with short tail? Okay. So this is very similar to what you did on the quiz. I hope you remember. So, go ahead, try it out. Let me restart the program. It's a little bit laggy. Okay, give me a second. Okay, Mohammed already got 140. Anyone else? Anyone else have another answer for me before I take it up? Or you guys need more time? All right. So the gametes for this one is big G, big L, big G, small g, big L, small g, small l. And you can find the gametes by doing this, right? Similar to what you do in math. And over here, we can do the same thing, but it turns out there are only two kinds of gametes, right? Small g, big L, small g, small l. So you're going to go ahead and do the Punnett square. And you can put the gametes on the side here. Okay. So hopefully you are able to do this. And, if you need a refresher, that quiz number 4 review video is still good for the purpose of preparing for the test. And, for you to do corrections on your quiz, right? If you needed to. And so over here, we are looking for gray with short tail. So gray with short tail, gray is the dominant trait, right? So anything with a big G, and then short tail has to be little l, little l. So actually, it's just this one. That is big G, which is gray, and then little l, little l. That's the only one that has what we're looking for. So that's 1 out of the 8 boxes. And we're going to multiply by the total number of mice. So 750 divided by 8, which is 93.75%. Sorry, 93. Well, this is 93.75%. But when you times 750, no. I'm sorry. Let me repeat that. Okay, 1/8 is 12.5%. When you take 12.5% times 750, you would expect to have about 93, right? 93.75, right? So 93 to 94, right? Mice. With the feature that we're looking for, right? That is going to be gray and short tail. So this is an example of a dihybrid cross. All right. You will definitely need to know how to do dihybrid cross on the test. When can you reuse the 9331? Yeah, you can only use the 9331 when the parents are heterozygous for both trait. So like if you're doing big G, big L, small l, cross another big G, big L, small l, then you can use the 9331 ratio to do it. Okay. Otherwise, you have to create your own Punnett square. Right? So in other words, the only time you use the 9331 is when your table is like 4 by 4, right? Then you can do it quickly. That's a good question. Anything else you guys want to ask me related to this? Now is your chance. Nope. Okay. Moving on. How many unique gametes? Oops. How many unique gametes could be produced through independent assortment? Right, so we did something like this in class. So basically you look at each gamete, right? If it's heterozygous, then you have two possible combinations. You can either pass on the big A or the small a. Right. 2 here. 1 option. Right? If it's big C, then you really only have one choice. You pass on only big C, right? And then same thing for the next one here. 2 here and 2 here. And you multiply all these together. So 16. Okay. 16. Consider a cat with long tail breeds with a cat with no tail. All the kittens have short tail. What inheritance pattern best explain this? Okay, so you get the heterozygous condition is somewhere in between, right? So when you have something that's somewhere in between rather than something that's like both expressed equally, this is called incomplete. Incomplete dominance. Incomplete dominance. All right, next one. People usually get confused. Will codominance be wrong? Yes, codominance would be wrong in this case because codominance means you have both trait, right? So it's not possible to have, like if you have a long cat and if you have a long tail and no tail, like you can't have both, or you either have tail or not have tail, right? It's not possible for both to show up, right? That's what codominance means. Codominance means both will show up, right? So you either have it or you don't have it, right? It's not like you can, in this case, it doesn't make sense to have both. So think of like the colors of the flowers I talk about, red and white. Codominance would be red petals with white spots on it, right? Whereas incomplete dominance would be like pink, like somewhere in between, right? Brown eyes is dominant over blue eyes. Is it possible for two people with brown eyes to have a blue eye child? Right, let's do a poll. Let's see what you guys think. Okay. If you think it's yes, then you choose A. If you think it's no, then you choose B. Go. Okay. Most of you think is yes, which is correct. Okay, because brown eyes is possible for B to be big B small b and big B small b, right? Like obviously if they're big B, big B, big B, big B, then it's not possible because everybody's gonna be brown eyes. But if they're heterozygous, yeah, then sure. Right? 25% chance. Blue eye. Okay. So you gotta think all the possibilities for a brown eye individual. All right, this is another one that's worth thinking about. So like on the test, right, you might have some multiple choice question that are like this. One person is brown eye, the other is brown eye. Which of the following is not a possible phenotype for their child? Okay. Similarly, Janet has normal vision, her husband has normal vision. Which of the following is not a possible scenario for the kids, right? So you really got to understand this. So let's talk about this. Janet is a female name. If you're not sure on the test, you can always ask me. So Janet is normal vision. Okay, so we don't know if she's like, XXC or XX. We really have no idea. And then for the husband, whom has no name, he has normal vision, so he must be XY. And there is just simply no other way. Okay. So let's see. Is it possible for them to have two sons where one has normal vision and then the other is colorblind? Is it possible? Yeah, it's possible. Okay, I don't know. Let's find out. Okay, so if one of the son is colorblind, then the mother must be XXC, right? So XXC cross XY. Okay. XXC cross XY. If you do the Punnett square for this case, right, you will see XX, XY, XXC, XCY. Right? So look at the sun, right? It's possible. Right? In fact, if you consider all the sun, then 50% chance the sun is going to be colorblind and 50% chance the sun is not. So yes, right? If Janet is a carrier, right? So on the test, you will have to explain to me that if Janet is a carrier, and then you show me the Punnett square, that 50% chance that the sons are going to be colorblind and the other is not, right? How about this? Is it possible for them to have two daughters where one has normal vision and then the other is colorblind? So if you look at the daughters, in this case, none of them is going to be colorblind, right? It's not possible, right? Sure, one can be a carrier, but carrier doesn't make you colorblind, right? So in this case is no. Okay. The only way for this to be true is if the father has color blindness as well, right? Okay. So if the father, basically, if the father is not colorblind, there's no way for any of the daughters to be colorblind, even if the mother is colorblind, right? Okay. Let me say that one more time, man. I want you to think about it. If the father is not colorblind, then the daughters are guaranteed to be not colorblind, regardless of the mother's genotype. Okay, because the father to have a daughter, the father needs to pass on the X, right? Okay, and if the father does not have colorblind, he would have passed on the normal X. Okay, which would override, which would cover up any XC that might be coming from the mother, right? Okay. So things to think about. All right. So this is what you were saying, right, Mohammed? That when we can use the 9-3-3-1. Consider two traits: flower color and the number of petals. Okay, flower colors and the number of petals. P generation. Pure breed. Pink flower with 6 petals. Okay. And then we have orange flowers with 4 petals. So basically we have two traits here. Pink, orange, that's color flower color. And then with 6 petals or 4 petals. So in F1 generation, 100% are orange flowers with 6 petals. So orange is dominant over pink. Okay. When I write like greater than like that, that means like dominant, okay? And then 6 petals is greater than, it's like dominant over 4 petals. Okay. So you cross F1 individuals. F1 individuals are all like, you know, big C, small c, big P, small p. Heterozygous for color and heterozygous for petal numbers. Okay. So if we take two F1 that are heterozygous for both of these, then you're gonna have a 4x4 Punnett square, right? Okay. How many will have orange flowers with 6 petals? Okay. And then here is a reference chart. Again, it doesn't have to necessarily match up with the letters that you're using, right? This is just for your reference. If you forgot what ratio represent what, right? So in this particular case, we are looking for orange flower. So that is a dominant trait, and then 6 petals. That is also a dominant trait. So if you forgot, I'm just gonna write it down for you. The 9 is the double D, right? Dominant and dominant. And then if it's dominant recessive or recessive dominant, those are 3 out of 16. But if it's recessive, the super rare one, that's only 1 out of 16. Now here we're looking for the dominant and the dominant. So you will do 9 out of 16 times 2,000. You know, we just plug that in. And that gives you 1125. Okay, so this table is just for you to use if you forgot. Okay, so dominant and dominant, that's gonna be these ones, right? Okay, the ones that I'm coloring, they are all, oh, sorry, I gave you the wrong chart here, actually. Sorry. I just realized it. That's the wrong chart. It should be something like this instead. Hold on, give me a second. It should be coming. There it is. Okay. It should be this chart. Okay, so in this case, you know, you're gonna have the dominant and dominant. Right, that's these ones. Right, so that's like the 9 out of the 16, right? Okay, so if you forgot, then you can make the chart, but it takes a long time, right? It's better to just remember the ratio. Yeah, thank you for pointing that out, Mohammed. I just realized it. Yeah. So are we okay with this? Again, you have to do these things on the test. Like, sure, you have some choice for question 4 and 5, right? 4A or 4B. and then 5A and 5B, but pretty much you have to know these just to be safe. Okay. All right, here's another one. We did this in class, but let's go through it again. Consider the cross between those two things. What is the probability of producing a genotype of that? Okay, so when we have more than 2 traits, is best to do each one separately. Otherwise, you have a really big Punnett square, right? So let's consider just the A. Okay, so big A, small a, cross big A, big A. And you need to do the Punnett square and figure out the probability of big A, big A. So we do this. All right. So probability of big A is going to be 1/2, 50%. Next, we're going to do the B. So consider big B, small b, cross big B, small b. You should be very familiar with these kind of tables now. And we're looking for big B, small b, right? That would be 1/2 as well. Okay. Just gonna highlight them for you. And then for the last trait, it is big C, small c, cross small c, small c. And we are looking for big C, small c, which is also 50%. Okay. So in total is half times half times half, which is 1/8. Okay. That's how you do it. Okay. There's no need to do like a big table. If you find yourself doing the big table, you're doing it wrong. Okay, let's take a look at this question. We have to create a pedigree. Hemophilia, which I told you about, it's a bleeding disorder, but it's an X-linked recessive. Use the following information to create a pedigree. So we can say X is normal, XH is hemophilia gene, right? Instead of XC, right? For colorblind, we use XH for hemophilia. So Alice has hemophilia. So this is Alice. Because she has hemophilia, she must be shaded. Okay. XH, XH. And she marries Terry. Terry is normal. And for male, normal, there's only one genotype. It's XY. Alice and Terry have 2 daughters and a son. Daughter, son. one of the daughters marry a man and they have a hemophiliac son and a hemophiliac daughter. Okay, so like I'm just gonna choose this daughter, it doesn't really matter. Marries a man, and then they have a hemophiliac son and a hemophiliac daughter. Okay. What is the probability that their next son also has hemophilia? Okay, so let's do the Punnett square for Alice and Terry, just to see what kind of combinations we can have. So XH for Alice and just XY for Terry. And then we can combine the letters. So this is the daughter here. Over here, that's the daughter. And looking at the Punnett square, there's only one kind of daughter that they can produce, and that would be like a carrier daughter. Okay, so this is guaranteed to be half shade in here. And same thing for her sister, right? She has to be a carrier. There's no other way. And for the son, right? If we look at the Punnett square, the son is guaranteed to be colorblind. Okay. Now let's take a look at the third generation. We have a colorblind granddaughter, so that's XHXH, and then the son here is also colorblind, right? So if you trace this back, one of the XH has to come from the mother, and of course the other one has to come from the father. Oops, that didn't work. Try again. Computer is very sluggish. This morning it's I haven't restarted for like a couple weeks. I think that's the problem. Let me see if I can fix this thing. Okay, so like this other one has to come from the dad, right? Okay. So for that, based on that logic, the father is also colorblind in this case. Okay. So now that we have figured out the genotype for these people, we can go ahead and do another Punnett square. And the question asks, what is the probability that their next son also has hemophilia? So we look at the sons only. Okay. Looking at the son, one of them is going to be okay, one of them is not going to be okay. So 50% chance. Okay. We say hemophiliac for adjective. Okay. So 50%. That's how you do it. Any questions? All right, remember, everything on this review sheet is going to show up on the test, one form or the other. Okay, nothing here is fillers. Nothing here is like, I'm just like showing you just for fun, okay? Like everything is going to show up. So you must know how to do, pedigrees, X-lanes, and that kind of stuff, right? Okay, let's go through this one. Answer each of the following questions. How many autosomal chromosomes are found in a normal human gametes? Okay, autosomal chromosomes or autosomes, these are the chromosomes excluding the sex chromosome, right? So a human gametes has half the genetic material. So that's 46 divided by 2, which gives you 23. And then we are only counting the autosomes. So that's 22. Okay. So half, and then take away the X or the Y, and then you get 22. How many chromosomes are found in a normal human cell that is in prophase? So look, right? This is the chromosome 1, and then chromosome 1, and then this is considered as 2, right? So after DNA replication, chromosome 1, 1, 1, 1, which is in prophase now. Okay, this whole thing is still counted as one chromosome and one chromosome. As long as they're stuck together, then it's considered as one chromosome. So if you started with 46 chromosomes, in prophase, it is still 46. Even we doubled it, but then since they're still stuck together, the way that we count them is the same. How many chromosomes are found in normal human cells during anaphase? In anaphase, we have separated the chromosomes. Now you pull apart the sister chromatids, so now it is going to be double. Okay, as soon as you pull them apart, then we count them separately. Even though it's like the same amount of genetic material, but if they're separate, count them as 2. Like yesterday, my daughter asked me for a cookie, and I gave her a cookie, and she's like, "I want 2." Then I took the cookie and then I break it in half and say, "Now you have 2." Right? She didn't quite understand. I was trying to mock her, but it's the same concept here. When they're together, it counts as 1, but when you break it apart, it counts as 2, just like the chromosomes. How many sex chromosomes are found in a normal human zygote? Zygote is what you will get after the sperm merges with the egg. So sperm has 23, eggs has 23, merged together you have 46, right? But now they're only asking for the sex chromosome. So the egg will always be carrying an X and the sperm will carry X or Y. When you come together, there will be 2 sex chromosomes in the zygote, XX or XY. How many sex chromosomes are found in normal human gametes? Just 1. Okay, so this is XX or XY. This one is X or Y. Okay. How many chromosomes are found in the cells of an individual with trisomy 18? Trisomy means an extra chromosome, right? In this case, extra chromosome 18. So normal cells have 46 chromosomes, but with the extra chromosome is 46 plus 1, which is 47. All right. Moving on. An organism has chromosome number of 2n equals to 12. So let's take a closer look at what these number means before you even look at the questions, okay? It's better to clearly understand this first. And you should do this on the test as well if you see something like this. So what does the 2 mean? 2 is the set number. If it's 2n, that means you have 2 sets. You are diploid. If it's 4n, then the thing has 4 sets, tetraploid. Okay, so it tells you how many sets you have. Remember, only even number of sets of chromosomes allows meiosis to take place. Okay, so 2n, 4n, 6n. If it's odd number of sets, then the organism cannot undergo meiosis. This here is the total number of chromosomes, and n is the number of chromosomes per set. Okay, you got to understand the difference between the set number and the number per set. Number per set can be odd number, even number. It doesn't matter. It's not going to affect whether you can do meiosis or not. Okay, but the set number, that's the one, like this, that's the one that determines whether you can do meiosis or not. Okay, so in this case, you got to do a little bit of math, right? N equals to 6 for this case, right? 12 divided by 2, it's 6. Okay, let's take a look at the question then. It says the organism is haploid, diploid, tetraploid. What do you think? Well, actually it says right here that it is diploid because it has 2, right? If it's 3, then it will be triploid. If it's 4, then it's tetraploid. This is just 2, so it's a diploid. How many sets of chromosomes does a somatic cell have? Okay, somatic cell is like bodily cell. That would be 2 sets. If they ask you how many sets of chromosome does a gamete have? A gamete is only 1, half of it, right? Germ cells is also 2, right? Because germ cells haven't undergo meiosis yet, right? Once it becomes gamete, then it's half the original set number. How many chromosomes does the cell have? 12. When we roll it down, total number is 12 right here. Okay. How many homologous chromosomes pair during, how many homologous chromosome pair does the organism have? Okay. So it has 2 sets. Each set has 6, right? So 6 pairs. Just like we have 23 pairs, right? How many chromosomes does the gamete of this organism have? 6. Half of the total. 12 divided by 6. 2 is 6. How many chromosomes does a cell undergoing metaphase have? Okay, so during metaphase, everything lines up in the middle. They have not separated yet. So it is still 12. All right. That's it. That's it for lecture 8. I just have to ask again. Anybody have any questions before I move on? All right. Biotechnology. We just have a couple of questions, and then body tissues should be okay. Okay, why don't we just take a short break, okay? Like, a couple minutes just to stretch our legs, okay? We'll come back at like 9:50, and then we'll do the rest, okay? If you think of other questions you want me to go through, just type them in the chat, all right? So I'll see you at 9:50. Can I explain the last question one more time? This one? Like the whole thing? Which part are you confused about? C? How many chromosomes does the cell have? So 2n equals to 12 means like 2 sets. And then the 12 is all the chromosomes, right? So like this number is already all of it. Okay. So why don't I give you another one for you guys to try? Okay. Let's try another one. And see. Let's say I have like, 3n. 3n equals to 15. Okay. 3n equals to 15. It's not possible for 3n to be 16. If 3n equals 16, then you have incomplete sets, right? You have like decimals, right, for sets, right? Okay, so try this. Right? We'll resume in a couple minutes. All right, let's keep going. Let me turn off the video and see if it's gonna make things a little bit better. You don't need to see me. So 3n equals to 15, right? Let's write down what this means for us. Okay, 3n equals 15. So n equals 5, right? N equals 5. So the organism, is it haploid, diploid, tetraploid? Well, if it's 3 means there are 3 sets. So we call this like triploid. All right. TRI is 3, right? Like you learn these prefixes in chemistry, right? When you name the chemicals. How many sets of chromosomes does a somatic cell have? In this case, it is going to be 3. Remember, this is the set number. How many chromosomes does it have? Just 15. Homologous chromosome pair does the organism have? Now, this wouldn't really be appropriate because it's no longer homologous pair, right? Because we have 3 sets, so it's more like homologous triplets, right? Like on the test, I'm like obviously I'm gonna rephrase it accordingly, right? So this one would have 5 triplets. Okay, so 1, 1, 1, 2, 2, 2, 3, 3, 3, like that. How many chromosomes does the gamete of this organism have? Not possible. Why is it not possible? Because it has odd number of sets of chromosomes. Therefore, it's not possible to undergo meiosis. No meiosis, no gametes, no sexual reproduction for this thing. How many chromosomes does a cell undergoing metaphase have? Same thing, 15. Any follow-up questions from anyone? Okay, if you're confused about this, you should ask. It's not going to go away, right? B, how many sets of chromosome does a somatic cell have? It's just the number in front of n. Okay, this is set number. That number tells you how many sets you have. A set is like, can I draw it? I guess. So like, can I draw it? Okay, yeah, sure. 1, 1, 1. Okay, that's first chromosome 1. 2, 2, 2, chromosome 2. 3, 3, 3. 4, 4, 4. 5, 5, 5, like that. Okay, great. I guess like as an analogy, right? Like if you think about setting up table for like a family dinner, right? How many sets of we call them culinary or like utensil, right? How many utensils do you need, right? Like, fork, knife, spoon. That's one set. Okay. So like if you have like 5 guesses, then you need to have 5 sets, right? Fork, 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 spoon, 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 like that, right? So like maybe that's gonna help you visualize what exactly is going on. Right? In that analogy, the fork would be chromosome 1, the spoon would be chromosome 2, and the knife would be chromosome 3. Kind of like that analogy. I just came up with it. I think I'm going to use it in the future. Okay, biotechnology, let's go. What is the purpose of PCR? The purpose of PCR is to basically clone and amplify DNA. You make many, many, many copies of like a specific sequence of DNA. If you start with 50 copies of DNA, how many would you have after 10 cycles? That's 50 multiplied by 2 to the exponent 10. Okay. Always 2 at the bottom. And then the exponent is the cycle number, how many times you do it. And then this is initial number of copies. Okay, so bring your calculator to the test. You're going to need them. 51200. That is all. Okay, how does it work? Remember, there is denaturation. Now you denature it. This is high temperature. You separate a DNA strand. And then you have annealing. Okay, lower temperature a little bit. And then you have elongation. Okay, which is a little bit higher temperature again. Okay, but not as high as the denatured temperature. Okay, you don't have to describe it, you just have to understand. Like if I ask you why do we need Taq polymerase, then you tell me because we have to heat it up to very high temperature to denature the DNA. And that's why you need to have an enzyme that is able to withstand the high temperature. It's not going to break down, right? Okay. Over here, we have what is restriction enzymes, which enzymes listed below create sticky ends, blunt ends, which one are more useful. So restriction enzymes are necessary for bacteria to defend themselves from foreign genetic material, right? That's essentially what they are. It's not something that's created by humans, right? Like this is something that's naturally occurring in bacteria, and they use it as a defense mechanism to protect themselves from foreign genetic material. Okay? So we extract them from these different bacteria, and sometimes it cuts zigzag, sometimes it cuts straight, right? So this is sticky end, this is blunt end again, sticky end, sticky end. Okay, and which are more useful? I should rephrase the question, which are more useful when it comes to inserting genes into plasmid? Right? Blunt ends ones are useful for other reasons that we didn't talk about. Okay, so these are useful for inserting gene into plasmid. As long as you cut the gene and the plasmid with the same restriction enzyme, then you will create complementary sticky ends. You can then glue them together using ligase. So over here, we have consider the following DNA sample, and we're cutting it with E-core R1. So equal R1 is GAATTC. I will always give you the table if you need to use it. So we just look for GAATTC, which is right here. It's one cut. And I don't see anything else. That's all. Right. And then we have SMA1. Okay, so look at SMA1 is GGGCCC. So we look for GGGCCC. It's right here. There is the cut. And that's the only cut. Okay, this time is blunt end. Sample 3, treated with ALU1. ALU1 is AGCT. So we look for AGCT. AGCT. There it is, AGCT. There's the cut. I don't think there is another AGCT. If you see one, let me know. Okay, and then we're gonna do equal R1 with small one. It's the same sequence that we've been using, right? So like you're gonna have a cut here by small one, and then we have the other cut here by equal R1. Okay, then we're gonna have to be able to draw the result of what it looks like when you load it onto a gel, right? So let me just, oh, that's totally gonna mess it up. Okay, hold on. Let's see if I can fix this. No, I cannot put them side by side. We're just gonna have to do it like this. Okay, I'm gonna make it a little bit smaller. Actually, can I do them side by side? Let's see, put it here. Oh, that's okay. There we go. So let's see. Lane number 1. We have just equal R1. So we have 2 fragments here. One is longer than the other. So never put it super close to the gel, to the well, because you don't know, like in another lane, if you have something that's actually longer, right? So just to play it safe, leave some space. So there is that, and then you have a shorter one at the bottom, right? Okay, so this is equal R1 only. Equal R1. All right, next we're gonna do small one, SMA1. And we have 2 cuts. And I'm gonna compare this fragment here. And that's like 1, 2, 3, 4, 5, 6, 7, 8. I'm gonna compare to this one here, which is like 1, 2, 3, 4, 5, 6, 7, 8, 9. And then it has a little bit like a longer thing here. So they are similar in size, but slightly shorter. So I'm gonna put it like a little bit further down. Okay, and then the other one is going to be a little bit higher up. Okay, this is SMA1. Okay, I mean, if this is shorter, then this one is going to be longer, right? So that's why one gets higher compared to the equal R1, and the other is lower down. Okay, and then we have the ALU1, ALU1. And it's just a cut like right down the middle. So these 2 pieces are both relatively long, but definitely shorter than before. So I'm just gonna put them somewhere here in between. And finally, we have equal R1 plus small 1. Okay, so bring some highlighters if you want to help you keep track of the gel. So like for example, over here, just waiting for the program to work. Is it working now? Okay, there it is. So there is a fragment, and it's the same fragment here, right? So I would expect to have the same piece, right? Using the equal R1 right there. And similarly, for the other side, right, this piece is going to be same as that piece. Okay, so you're going to have another one down here. And then you're going to have like a middle piece here. Okay, so this middle piece is probably somewhere here. That's it. That's how you do the gel. Okay, so practice this gel question, and then we had another one in the study guide. Okay, make sure you understand the theory behind it. Okay, just a couple more questions here. Give an example of a palindromic sequence that is 6 nucleotides long. That's really straightforward. You just make up any letters that you like. If it needs to be 6 long, then I do half of that. Okay, I make up any 3 letters. So like, you know, A, C, G. And then you start to halfway through, you start to do the complementary. So complementary to G is C, complementary to C is G, and complementary to A is T. That's it. Okay, and just to be sure, you can do the complementary strand on the other side, just to double check that it's the same. Yeah, GGGCCC also works, right? If you want to keep it simple. Can I go through the blunt end and sticky end again? Like, as in, like, how are they different, or what do you mean? Okay, how are they different? Okay, it's different based on how it's being cut, right? Like if it's cut in a way that is zigzag like this, okay, then you have sticky ends, complementary. AATT is complementary to TTAA. Blunt end means you cut straight down. So there's no, we call these like overhangs that allows them to come back together. But if you do it straight down, there are no overhangs. They cannot come back together. So for the purpose of sticking DNA ends together, you really want to have an enzyme that creates these sticky ends. Okay, three different DNA samples are treated with equal R1 and separated using gel electrophoresis. What is the purpose of the marker lane? Okay, the marker lane provides a reference for DNA length. Okay, these are preloaded DNA in that comes in a tube, and it basically allows you to measure the sample, right? Which sample has the longest DNA strand before being treated with equal R1? Okay, so let's take a look at A. Sample A. That one, if you compare it to the marker, is 1,000. Okay, I only see one band. And then for B, we have this one is 800. This one is 200. 800 plus 200 is also 1,000. Over here, we have, you just read over, right? 600 plus 300 plus 100, which is also 1,000. So before being treated with equal R1, they were all the same size. Okay, they're all 1,000 to begin with. Which sample was not cut by the restriction enzyme? Sample A, right? Sample A. Only one band. Okay, assuming these are not plasmids, right? Okay. I mean, there is another assumption that we make. I mean, you don't have to know this on the test, but like I'm just telling you, okay, just in case you're wondering. Let's say this is the DNA sample, right? If you cut it right in the middle, then you're gonna also only have one band, right? So we are assuming that didn't happen here. Okay, maybe just for the sake of argument, right? Maybe the thing was 2,000 base pair long, and then you cut it exactly in the middle. Then you're gonna have, you know, just a 1,000 band, right? Okay, but we are assuming that didn't happen here. All right. I think we talked about this already on the test, on the study guide, but I can quickly go through it again. If you forgot to add the loading dye, that's the blue stuff, then the DNA will not sink into the well. They will just come back up into the buffer, and sample would be gone. Okay. If you reversed the positive negative terminal, the gel will be running the other way towards the well, as supposed to be away from the well. You will lose the sample. You left the power supply for too long, you will also lose the sample. It will all exit on the other end, right? So basically, ABC, you will all lose the sample, but in different ways. If you forgot to add ethidium bromide, you cannot visualize the DNA. You cannot see it under UV light. Okay, only the last one can be corrected. Okay, first three, if you made a mistake, there's nothing you can do to correct. You got to restart your experiment. Okay, but if you forgot to add ethidium bromide, that's okay. You can just add it and then soak the gel again, and it'll be done. Can I explain? Yeah. The loading dye. You remember the loading dye? It's the blue stuff, right? That stuff is really thick. So let me draw the gel for you. This is the gel. Okay, gel is like a 3D slab, right? Okay, so this is the well. The well is literally like a slot, okay? So like when you add the DNA sample, the dye would help the sample sink into the gel. So that the DNA is actually gonna be like running in the gel, within the gel. If you didn't add the dye, then you squirt the DNA and the DNA is just gonna be in the surface, and then it will just diffuse back out into the buffer and be gone. It didn't sink into the gel. Basically, that's what it means. Does that make sense? Still a little bit. Okay, all right. There are two DNA samples with STR. STR stands for short tandem repeats. Polymorphism. We did this in class, but I thought you needed a refresher. This one is 3 nucleotides repeated 10 times, so that's 30 in total. This is 3 nucleotides repeated 15 times, so that's 45 nucleotides in total. Which sample will be further down the gel when separated using gel electrophoresis? GAC, nope, copied it wrong, sorry. CAG rather, 10 is shorter. Okay, therefore, further down the gel. The shorter the DNA, the easier it is to weave through the gel. Okay, and therefore further down it goes. All right, over here, we did this many times already. The plasma below is treated with EcoR1 and BAMH. When the digested plasma is separated using gel electrophoresis, how many bands will you see? If you cut EcoR1 here and BAMH, you will have 2 bands. 1 band, 2 band. Okay, the one on the left will be closer to the well. and then the one on the right would be further down because it's like shorter, right? A gene is inserted into the same plasmid shown in question A using equal R1. The plasmid is transformed into a tube of E. coli. You culture all the E. coli on the plate using tetracycline. What will happen? Again, you should be familiar with this now. If you insert using equal R1, right? This does not disrupt the TETR gene in the plasmid. Okay, if it does not disrupt the tetracycline resistant gene in the plasmid, then any bacteria that has accepted the plasmid is going to have tetracycline resistance. Okay, so any bacteria that accepted the plasmid will grow. In the presence of tetracycline, right? Will the outcome be different if you insert it using small one? Yes, it will be different for sure. Small one is gonna cut right in the middle of tetracycline resistant gene, and even if you accepted the plasmid, it's not gonna help. It's gonna die. Okay. So the question you have to ask yourself on the test is whether or not the insertion is going to be disturbing the resistant gene. All right. No exclusion for this lecture. You have to know everything. Sorry. I think that's about it. I mean, I'm not gonna go through these things again. I think you guys are pretty okay with the body tissue. All I am gonna say is for the body stuff for lecture 10, let me see. 1, 2, 3, 4, 5, 6, 7. There are only 7 questions on lecture 10. 7 MC. Okay, it's not going to show up in the multiple choice. It's not going to show up in the written section. It's all going to be multiple choice, and it's going to be like the questions we did in class. Okay, where I asked you things like the fingers are blank to the wrist, and you have to choose the appropriate term. Okay, so you should be okay. It's pretty straightforward. Okay? This is not the lecture you should be worried about. The one that you should be spending most of the time understanding and studying is the the 8 and the 9. Okay, those are the hardest lecture of the whole course. All right, anything else before I go? Any other concepts you want me to review? Doesn't even have to be 8, 9. It could be like from earlier, 5 or 6. Well, not 5. 5 is not an exam. I mean like microscope and 6. Anything at all? Okay, well, you know where to find me if you have any questions. You can just email me. Can I give the answers of last question in notes? What you mean these ones? I mean it's not really answers though, right? Like this here, this is straight the picture directly from the PowerPoint. The key thing to know from this picture is what is in each of the cavities. For example, we did this in class, okay? This is the thoracic cavity. Okay. So give me something that is in the thoracic cavity. We did this last week, I think. Anybody? What's in the thoracic cavity? Yep. Questions right. There's lungs. And then one more. Heart. That's it. Okay, so just know some of the stuff. And again, I'm not gonna give you like weird things. Okay, I'm just gonna give you things that are obvious. Sadie, who are you anyways? What, that's not your name, right? Like you just using someone else's login? Is that because I don't recognize that name for my class? For the palindromic sequence, it's yeah, GGGCCC is fine. That's right. Oh, that's okay. Yeah, great. Oh, is that what you go by? You actually go by that name instead of the full name? Okay, you could have told me. We've known each other for like the whole semester, and I found out in the last class. In any case, so that's that. Give examples of these terms, right? So I mean, there's no answer here, right? Like you just kind of have to know how to use them. All right, that's it. I will see you all on Wednesday in class. Good luck. I'll post the notes as well as the recordings online as soon as they're ready. Probably like in, half an hour or so. Okay, good luck, guys. Bye for now.